Answer briefly. So students, now here 4 questions are there. Let them answer. So before answering, let us see what the questions are. Why are villi present in the intestine and not in the stomach? So the second question says, how does pepsinogen change into its active form? The third question is, what are the basic layers of the wall of alimentary canal? How does bile help in the digestion of fats? Now these are the 4 questions. Now let us learn the answer of these questions. The first one, why are villi present in the intestine and not in the stomach? So students, here we can observe a small finger-like projections. So these finger-like projection is called villi. So here is an enlarged one, a single finger-like projection. Now this entire thing is called villi, and these are present in the small intestine and they're not found in the stomach. Why? So let us see. So before that, we'll just briefly observe the structure of the villi. So here each villi contains a lymphatic vessels, which is green in color, blood vessels, that is arteries, veins and capillaries, and surrounding that we have number of cells, that is epithelial cells, and at the end of the epithelial we can see projections called microvilli, which increases the surface area. Now this is the structure of the villi. So this is found in the intestine. The mucosal wall of the small intestine forms a million of tiny finger-like projection known as villi. So here inside the wall of the small intestine we can find a million of tiny finger-like projection. So each projection is called villi. So when there is a repeated finger-like projection formed, each one is increasing the surface area for the process of digestion and absorption. So the villi increases the surface area for more efficient of food absorption. So we know that in the small intestine the food gets completely digested. So the nutrients should be absorbed. It is taking place in the lumen, right? So when the wall of the intestine undergoes many finger-like projections, they are in turn actually increasing the surface area for the absorption of the nutrients. Now that is why a villi has got many blood vessels in it. So inside, if this is a villi, inside it there is a presence of the blood capillaries. Arteries, veins and capillaries are present. Now the digested food is taken into the blood, and through this blood it will be transported to the other cells in the body. So along with that there is the lymphatic vessels. This lymphatic vessels helps in the absorption of the fats. So in the small intestine, carbohydrates, proteins, fats, all the 3 gets digested, but the blood vessels can absorb proteins and carbohydrates, whereas the lymphatic vessels can absorb the fat. Now all these nutrients which is absorbed will be carried to the different parts of the body by the lymphatic vessels and the blood vessels. The question is, why was this villi present in the intestine and why it is not found in the stomach? So even the wall of the stomach has also irregular foldings. This folding is known as rugae. So here we can see the folding of the wall of the stomach, and this folding is called as rugae. So this increases the surface area to the volume ratio for the expanding of the stomach. So we can observe here it helps in expanding. So it helps to expand the stomach so that the food which is present inside will be stored for few hours, that is 2 to 3 hours, and within this duration the digestion in the stomach will be completed. So rugae is not supplied with the blood vessels. Here it does not undergo absorption. It will just increase the surface area to volume ratio so that the stomach can expand and store the food. So villi is present in the intestine for increasing the surface area for the absorption of the food, and that is why they are not found in the stomach. Now coming to the next question, that is, how does pepsinogen change into its active form? So students, generally any enzyme which is present in the inactive form. So here even the pepsinogen is present in the inactive form. If it is present in the active form, it can digest the proteins which is present in the wall of the stomach. So to avoid that, generally these enzymes are produced in the inactive form, and that inactive form is called pepsinogen. They get activated in the presence of the hydrochloric acid, HCl. When the pH reduces, when the pH becomes acidic, the pepsinogen gets activated and the pH reduces in the presence of HCl. So when the HCl is present, the pH of the stomach will become 2. So when the acidic pH is formed, the pepsinogen becomes into active form called pepsin, and thus it digests the proteins present in the stomach. And this occurs only when the food is taken in. So when the food enters into the stomach, HCl will be released, and this HCl converts the inactive pepsinogen into active pepsin. So pepsinogen is a precursor of the pepsin. Precursor means in the inactive form. It is stored in the wall of the stomach and it gets converted into its active form by the hydrochloric acid. So here we can see this is the gastric glands. So gastric glands have got cells, that is parietal cells, which secretes HCl, and this cell activates the pepsinogen into pepsin. So pepsinogen are present in the chief cells. So the chief cells releases pepsinogen, which gets activated into pepsin by the release of HCl. So HCls are released by the parietal cells. The next part of the question, what are the basic layers of the wall of the alimentary canal? So now let us learn the layers present in the walls of the alimentary canal. So walls of the alimentary canal has 4 layers. So they are serosa, muscularis, submucosa and mucosa. So these are the 4 different layers present in the wall of the intestine. So serosa, it is the outermost layer present in the alimentary canal. It is made up of epithelial cells. So serosa, since it is an outermost layer, we know that the outer layer generally consists of epithelial cells, and below it there is the presence of some connective tissue, because epithelial is avascular and most of the epithelial will be having an underlying connective tissue. Now this becomes the outermost layer. Now the second layer is muscularis. So the name itself says. So muscularis means the layer which is made up of muscles. So here muscularis is a thin layer of smooth muscles, that is they are involuntary in nature, and they are arranged in 2 layers, a longitudinal layer and inner circular layer. So here if you observe, there are 2 layers, the outer longitudinal layers, so these looks longitudinal, and inner one is it is present in the circular forms. So when we take a transverse section, we can see an outer longitudinal layer and inner circular layer. Together this layer forms muscularis. They both are smooth muscles, they are involuntary in nature, it is not under our conscious will. The next layer is submucosa. So this layer is the submucosa. It is made up of loose connective tissue. So it consists of nerves, blood and lymph. So it is a region which is supplied with the nerves, blood vessels and lymphatic vessels. So the nutrients which is absorbed or which is supplied to the cells will be taken by the blood, lymph, and the information will be carried by the nerves. Now this becomes, it supports the mucosa, which is the innermost layer. So mucosa is the innermost lining of the lumen, and it is mainly involved in the absorption and secretion. So it absorbs the nutrients after the digestion and it secretes the enzymes for the digestion. So totally there are 4 layers in the alimentary canal. Outer layer serosa made up of epithelial cells. Second layer muscularis made up of smooth muscles. Third layer submucosa, which is supplied with nerves, blood and lymphatic vessels. And innermost layer mucosa, it is mainly involved in the absorption and secretion. Coming to the last part of the question, how does bile help in digestion of the fats? So bile juice is secreted by the liver and the excess will be stored in the gallbladder. So this bile juice will be released into the intestine, and in the intestine the process takes place. It helps in the digestion of fat. So there are many roles for the bile juice. The first one is it reduces the acidity. So it is alkaline in nature because it has got bile salts in it. So once the bile juice is released, digested food from the stomach enters into the small intestine. And this digested food is called chyme because it is mixed with the acidic gastric juice. So to make it alkaline, the bile juice will be released. So bile, because of the bile salts, it is alkaline in nature. So it neutralizes the food which is coming from the stomach into the intestine. So that is one of the function. Second one, it helps in the digestion of the fats. So here we can observe these are the fat globules which are bigger in size. Once the bile salts acts upon the fat globules, it breaks this larger fat globules into smaller fat globules. So once the fat globule is broken down, the enzymes from the pancreas are in the small intestine, which helps in the fat digestion, like pancreatic lipase. They further break down the fats into its simpler form, that is fatty acids and glycerol. So this will be absorbed by the lymphatic vessels. So this bile helps in the digestion of the fat. It is not an enzyme. It does not directly digest the fat, but it breaks the larger fat globules into smaller globules. It breaks the larger fat globules into smaller globules so that the pancreatic enzymes can act upon it and break down into its simpler form. So this process is called as emulsification of fat. So these are the brief answers for the questions.